This is question 9 of the core concepts in class book, and we're on to nucleic acids. In 1961, a group of scientists demonstrated that the genetic code for a protein contained with DNA uses a triplet code. Okay, so we know all about triplet codes. How many different types of amino acid are used in protein synthesis? So AO1, remembering that there are about 20, 20, 21, depending on who you believe. Straightforward. Why there must be 3 bases in each code? Well, we've got our bases. How many different bases do we have? There's only 4 different bases, so there's only 4 different bases. A, T, G, C. And if they coded for 1 amino acid each, then we'd only be able to code for 4 amino acids. If we use a double system, then that still wouldn't get to our 20. If we had a double system, that would code for 16. And so we need a triple system to code for more than 16, so 20 there. So 1 or 2 bases do not code for enough amino acids. The 3 base system codes for up to 64 amino acids, so plenty to cover our 20 there. And here are some codons and the amino acid that they would code for. And we've got a section of messenger RNA produced in the nucleus. And that's there. So immediately what I tend to do is separate these into our triplets. So I would advise you to separate these into triplets almost immediately, and it helps you get things organised on the page. Using the information given in the table, write out the sequence of amino acids for which this section of messenger RNA codes. So traditionally students are pretty good at doing this. So AUG, that is methionine. Then GUU, valine. GAA, glutamic acid. So I want to get in GAU, aspartic acid. GUU, valine. And the last one, GAC. Here we go. Aspartic acid again. GAU, yeah, okay. Okay, write the base sequence for the DNA template from which the strand was produced. So A's go with T's, complementary base pairing. This is what would be a T there, so that would go with an A. And C's and G's, so C's wherever there's a G, and a G wherever there's a C. So we have got T, A, C, C, A, A, C, T, T, C, T, A, C, A, A, and C, T, G. So reasonably straightforward so far. Describe the process occurring in the nucleus that gives rise to this strand of messenger RNA. So we need 4 things here. So 4 separate points. Let's mention our first enzyme, DNA helicase, uncoils the DNA. We can say, breaks hydrogen bonds to separate those 2 strands, to separate strands. We've got our exposed DNA bases are a template, and then we're going to get a new strand of messenger RNA formed by complementary base pairing. Of free RNA nucleotides, or free RNA nucleotides, and we can mention RNA polymerase attaches nucleotides to the chain. So in there somewhere we just need to hit 4 separate good points. During DNA replication, a mutation occurred in the sequence of bases from which messenger RNA was produced. The mutated DNA resulted in messenger RNA with the following base sequence, and they've helpfully separated this into our 3s here. But less helpfully, they haven't given you all the information that you need to complete this question properly. Let's have a look at what they've given us here. AUG, that's the same. So that's still going to be the same as what we had before. GUA, that appears to be a mutation. Let's look in the table. GUA. GUU. It's not there. So this is a mistake in the paper. My Googling of messenger RNA codons suggests that GUA would also give us valine. Okay, but if you came across this question, you wouldn't necessarily know that. So that's going to produce the same amino acid. GAA, that's the same. GAC, oh okay, that's changed. So it was, so GAC was, let's have a look. GAC was aspartic acid, and GAU, oh, also aspartic acid. Oh, it's the same one as well. GUU, and that's valine, and that's the same, and GAC. So we did have a couple of changes here. One you could guess was the same one, but you can't tell from the table, and the other one, it's a different codon, but it's the same amino acid that is coded for. So it's kind of a part of our degenerate code where more than one codon is used for one amino acid. So we just need to put that into words. So the mutation GAU to GAC codes for the same amino acid. Which is aspartic acid. So, the same primary structure is made. So no changes. So 30 marks. Apologies from the exam board for that slight mistake. But this was I think it was a specimen sort of paper anyway. So they're not checked quite as well as the others.